Welcome to the video lecture for Unit 5, Module 10.2.2. In this one, we're going to continue our discussion of the small intestines by covering the bladder and talk about the coordination of secretion and absorption. As always, here are your learning objectives. Be sure to utilize these when studying for your lecture quiz and the unit exam. The gallbladder is a hollow pear-shaped organ that stores and concentrates bile prior to its excretion in the small intestines. This muscular sac is located in a fossa, or a recess, in the posterior surface of the liver's right lobe. The gallbladder is divided into three regions: 1, the fundus; 2, the body; and 3, the neck. The cystic duct extends from the gallbladder to the point where it unites with the common hepatic duct to form the common bile duct. At the duodenum, the common bile duct meets the pancreatic duct before emptying into a chamber called the duodenal ampulla, which receives buffers and enzymes from the pancreas and bile from the liver and gallbladder. The duodenal ampulla opens up into the duodenum at the duodenal papilla, a small mound. The muscular hepatopancreatic sphincter, or the sphincter of Oddi, encircles the lumen of the common bile duct and generally the pancreatic duct and the duodenal ampulla as well. A major function of the gallbladder is bile storage, but it is released in the duodenum only under the stimulation of the intestinal hormone CCK. Without CCK, the hepatopancreatic sphincter remains closed, so bile exiting the liver in the common hepatic duct cannot flow through the common bile duct and into the duodenum. Instead, it enters the cystic duct and is stored within the expandable gallbladder. Whenever chyme enters the duodenum, CCK is released, relaxing the hepatopancreatic sphincter and stimulating contractions of the gallbladder that push bile into the small intestine. The amount of CCK secreted increases markedly when the chyme contains large amounts of lipid. The gallbladder also functions in bile modification. When full, the gallbladder contains about 40 to 70 milliliters of bile. The composition of bile gradually changes as it remains in the gallbladder. Much of the water is absorbed, and the bile salts and other components of the bile become increasingly concentrated. If bile becomes too concentrated, crystals of insoluble minerals and salts begin to form. These deposits are called gallstones. Small gallstones are not a problem so long as they can be flushed down the bile duct and excreted. In cholecystitis, the gallstones are so large that they can damage the wall of the gallbladder or block the cystic duct or common bile duct. In that case, the gallbladder may need to be surgically removed. This does not seriously impair digestion because bile production continues at normal levels. However, the bile is more dilute, and its entry into the small intestine is not as closely tied to the arrival of food in the duodenum. A combination of neural and hormonal mechanisms coordinates the activity of the digestive glands. These regulatory mechanisms are centered on the duodenum, where acids must be neutralized and appropriate enzymes added. Neural mechanisms involving the CNS prepare digestive tract for activity through parasympathetic innervation or inhibit it through sympathetic innervation. Neural mechanisms also coordinate the movement of materials along the length of the digestive tract through the enterogastric, gastroenteric, and gastroileic reflexes. In addition, motor neurons synapsing in the digestive tract release a variety of neurotransmitters. Most of these chemicals are also released in the CNS. In general, their functions are poorly understood. Examples of neurotransmitters that may be important include substance P, echinophyllines, and endorphins. So we'll now summarize the information presented thus far on the regulation of intestinal and glandular function. We'll also consider some additional details about the regulatory mechanisms that involved, so we're basically combining the two modules that we had here. The intestinal tract secretes a variety of peptide hormones with similar chemical structures. Many of these hormones have multiple effects in several regions of the digestive tract and the accessory glandular organs as well. Other intestinal hormones are produced in relatively small quantities. Examples include motilin, which stimulates intestinal contractions, villikinin, which promotes the movement of villi and the associated lymph flow, and somatostatin, which inhibits gastric secretion. For slides 7 and 8, this just highlights the functional interactions among gastrin, GIP, secrin, CCK, and VIP, and they're diagrammed here. On average, it takes about 5 hours for materials to pass from the duodenum to the end of the ileum. The first of the materials to enter the duodenum after you eat breakfast may leave the small intestine at lunchtime. Along the way, the organ's absorptive effectiveness is enhanced by the fact that so much of the mucosa is movable. The microvilli can be moved by their supporting microfilaments, the individual villi by smooth muscle cells, groups of villi by the mucularis mucosae, and the circular folds by the mucularis mucosae and the mucularis externa. These movements stir and mix the intestinal contents, changing the environment around each epithelial cell from moment to moment.